This story began about 20 years ago. We were working on transplanting vascularized turkey tendons in our university research laboratory. And we were amazed by how complex the vascular network was. Then the Chinese School of Plastic Surgery introduced the concept of the retrograde flap, and we began developing the technique of vascularized retrograde tendon transfer, extensor transfer, or median bundle transfer to address the problem of flexor or tendon reconstruction. Gradually, this new vision of anatomy began to shed light on the interaction of tendons, sliding sheaths and vessels, and challenged the very foundations of classical anatomical thinking. During revascularization after Garrett removal, we noticed that sensory endings bled not only as a result of obvious circular, longitudinal and peripheral vascularization, but also contrary to the existing vascular theory at that time. It seemed that there was a real continuous connection between the sheath, tendon, and vessels, which did not break despite the stretching that occurred as a result of sliding. From observing the behavior of vessels in the common sheath during flexion-extension, it also became clear that simplified mechanical explanation could no longer be used to explain this phenomenon, and that this apparent disorder and irregularity of forms was actually the basis of some new kind of complexity, about which we probably still know very little. Trying to find an explanation, we began a detailed microscopic study of vessel mobility distribution using 25x optical magnification. Looking at the vessel as an analogy to a gas pump, we were able to approach solving the problem more simply. Here, this vertical structure looks like the main part of the pump, then comes the hose, and finally, the attached vessel like a car refueling at a gas station. The hose seems to stretch as the car drives away and folds when it approaches. Look at this sequence. The large vessel, vessel number 3, during flexion gradually moves behind vessel 2, the pump. Then it passes by it, reaching vessel 1, the car, the swelling on which gradually straightens as the hose stretches to its maximum length. And finally, vessel 3 overtakes vessel 2 and approaches vessel 1, which in turn moves away from vessel 2. As it appears in homogeneous living matter, there are several forms of speed and progression. Due to the limitations of nonlinear thinking, previously the only explanation was the idea that vascular structures were reinforced by several coaxial layers of connective tissue. The closest of the moving ones, the fastest, and the farthest, the slowest. This meant we needed a new way of thinking, something that presented problems from the perspective of global dynamic continuous matter. We needed a theory that included the concept of tissue continuum in complete contradiction to the traditional view of sliding structures, in short, including the concept of stratification and virtual space. Electron microscope observations after dehydration showed that this collagen network actually forms a continuous matrix consisting of millions of tiny disordered vacuoles, obviously organized in a fractal manner. These were pseudo-polygonal, but very special dynamic formations of volumetric glycogen, whose environment consisted of first, third, fourth, and sixth type collagen fibers. Significantly different in size depending on anatomical location and the role they were supposed to perform. In this chaotic random world, there was absolutely no geometric order or linearity, only fractal chaos. This system is traditionally known as connective or areolar tissue, but we called it in our note the microvascular collagen absorbing system. In our opinion, this was a rubber elastic system, because its role was to avoid reaching the resistance threshold at which collagen could separate. It should allow tendons to move freely without transmitting movement to surrounding structures. It also amazed us that the biomechanical behavior of the shock-absorbing system could be described by a sliding drawer model. As the tendon moves, the first vacuole must undergo attraction to the point where it initiates interaction with the next adjacent vacuole. Then the third, fourth, and so on are activated, so that the last vacuole remains practically inactive. Our second hypothesis was that the same process was involved, but pressure was distributed among the vacuoles equally. However, this meant that we were still within only 2 dimensions, and that our only explanation was based on linear processes. But there was something we believed in the tissue continuum, the existence of basic vacuolar-type structures, and the existence of a damping system that allows organs to act independently. We didn't need to move into the 3rd dimension to really understand what was happening. And this was at the time when computer image synthesis appeared. It allowed us to visualize structures as a system in constant motion with 2 rigid boundaries: fixed external and free mobile internal. What we did was to introduce the concept of interconnected network-type architectures, including microvacuoles on some framework. Then we looked for fractal distribution. Scientific approach and graphic modeling pointed to one and the same reality. It really was a sliding system with shimmering fluid located between the vacuoles and collagen fiber reinforcement. This confirmed what we initially thought about what happens between vacuoles during tension and stretching. Moreover, thanks to this discovery, we were able to confirm that the ability to move relative to each other is inherent not only to tendons. Entire organs can behave similarly, that is, this system was common to the whole body. It was then that we realized we had discovered that this microvacuolar collagen absorbing system is found everywhere in the body. It's sliding tissue in every nook and cranny of the body, in other tendons such as extensors, in the back of the abdomen, around the rectus abdominis muscle, in the area of contact with the rib cage, around the erector spinae muscle, in the reflecting conjunctival groove, and so on. It may actually appear that this tissue serves as an anchor for muscles and tendons, and may even be their primary constituent material. Even structures not intended to move as such, nerves and periosteum, consist of fibers of this same tissue, but with differences in their arrangement and size. The metascopic framework of the study required a more holistic approach for complete understanding. Thus, we began to consider skin and subcutaneous structures, which we thought would be more stable in this mobile tissue, the studied environment. By the structure of its role as a container for other sensory organs, skin is more than just an organ. It's a set of organs, complex anatomical, physiological, cultural, psychological. Of all the senses, touch is the most fundamental. You can survive without smell, sight, or taste, but not without touch. Skin constantly transmits information. It never turns off, doesn't block, and doesn't sleep. It has odor and texture.
It sécrète fluide, sécrète, filtre.
It exchanges signals with the outside world. Thanks to skin, the body surface is a communication machine, as well as their protective barriers. It can change color, texture, and shape, and resist aggressive influences such as sunburn, scars, and disease. At birth it's tight, ready for life, then gradually it ages, matures, forms folds, and sags. But neither in newborns, nor in teenagers, nor in adults will the skin ever be completely flat and smooth. Through the hair on its surface you can see a complex geometric structure, divided by straight lines, allowing movement in all directions of space. These triangular pyramidal structures move relative to each other. At any moment they align in the direction of tension. The previous arrangement disappears, and they take on a new form, then as if by memory they return to their original position. With every daily gesture, groomed or neglected, skin undergoes similar types of transformation, tension, stretching, but still with the exception of open wounds returns to its original state. Of course, the ability for such movement has been known from time immemorial. It's called elasticity, but the question is how this elasticity is connected to subcutaneous structures. Using a video endoscope, We were able to take a walk under the skin to try to understand this seemingly simple world of sliding, mobility, and smooth, slippery surfaces. We wanted to understand how it can combine the ability for fast, sometimes sharp movements, as well as the finest and gentlest without any jerks. What do we see when we make an incision with a surgical blade? Simply a wealth of tissue connections, histological continuum, without clear separation of skin, dermis, hypodermis, vessels, aponeurosis, and muscle. How then can all this move so smoothly? And how can sliding occur? Looking closer, you can see that directly under the layers of dermis and hypodermis lies an extremely mobile tissue that encompasses everything else and penetrates into what is called the undermining plane. If we pull this tissue upward, we get a surprise. As a result of the explosion in equolia under atmospheric pressure, an interesting movement will begin, demonstrating the existence of hydraulic systems at various pressure levels. This is the total action of fibrillar structures of the same geometric form, which encompasses muscles, skin, and fatty tissue. But what are these forms? What do they do and why are they here? Is this what is traditionally called unitary tissue? Let's dive into this world of fibers. Initial analysis reveals mainly two types of structures. Some are quite wide, with a knife-like edge and transparent surface. These surfaces consist of glycoprotein derivatives. Others resemble extended fibrils, which can be thin, long or short, swollen or cylindrical. At first glance they look similar, but look more carefully and you'll see enormous diversity. Bird plumage, reed stems, rush bundles, wheat grains, very resistant to stretching, ropes, rigging, cords with ring strengthening durability, like bamboo segments, transparent sails and dewdrops. Move along these pearlescent structures, and you'll notice the same fractal order everywhere. Large fibers, endlessly penetrated by small ones. Total tissue continuum of homogeneous dimensionality, completely fractal order. A world of chaos and fibers. The human body is built, it would seem, of one and the same tissue, which differentiated over time, beginning with basic stereotypical organization. Then this organizational structure, supporting life, must have its inherent behavioral rules. How are these structures organized? How do they absorb substances? And how do they move? And what role do they play? Using the language of physical mechanics, this structure can be called complex, completely stable and seeking equilibrium, a system with enormous ability to resist change and adapt to it. Neither physicists nor doctors know of another such entity that would produce so many cacophonous movements at microscopic and macroscopic levels, movements involving muscles, fluid, and cell fibrils. Here we are at the very heart of complexity. The combination of collagen based on glycogen with reservoirs of fatty microglobules is a repository of energy, information, and matter. The communication network for exchanging energy, substance, and information between system components and various reservoirs is the blood flow running between nerves, veins, arteries, and cells. Some mechanisms are still poorly studied, for example, pressure oscillation visible in these images, cable movement inside the thread, apparently hollow inside. And this is not the whole story. In this global network of encompassing skin, there are also valves capable of increasing or decreasing the flow of information in terms of intensity and speed. Storage time here plays a leading role in enhancing or inhibiting other mechanisms and directly affects fat and lymphatic reserves. This is a multi-microvascular system that at first glance seemed chaotic and complex, thus representing a set of elements with a large number of nonlinear interconnections, arranged with a universal purpose of promoting life, subdividing and multiplying according to predetermined criteria such as oxygen level, body temperature, and plasma composition. This internal state of equilibrium must be maintained at any cost. Nevertheless, this internal equilibrium includes another important aspect: maintaining structural equilibrium not from the perspective of flows or transmitters, but from the perspective of mechanical stability. To transmit anything, a structure must be able to resist, adapt to the basic requirements of the environment, and maintain its architecture. Most sequences demonstrate the existence of homotypic geometric figures with polygonal distribution. This visual impression is most simply expressed in its basic element, the microvacuole. We have already seen this in relation to the paratendinous system. It's arranged differently depending on the role it's called to play. The more longitudinal displacement, the smaller and more frequent the forms in the vacuolar organization. However, the basic structure of the polyhydride fibrous structure filled with gel remains the same. The point is that this network must provide its own overall mobility without affecting anything from the surroundings.
Dynamics must be absolute and damping impeccable.
These two seemingly contradictory requirements must, moreover, be complemented by a memory function of the original state while conserving energy and releasing heat. The rheological equation, which represents the local relationship between stress and deformation, can be considered from the perspective of elastic rubber-like deformation, as suggested by the model of collagen fibers, which cannot stretch infinitely. Another direction of research thought suggests that a mechanism based on ion concentration fluctuation should work here. The problem is still open, but we believe the key lies in the concept of total transmitted stress. Each fibrous element can be connected to its neighbor under some initial stress. When external force is applied, the adjacent element may experience slightly less stress until it reaches the required deformation. Nevertheless, these sequences of alternating, interweaving fibrillar structures created by repetition of movements within movements cannot be explained by standard reasoning. Physical laws using nonlinear mathematics are required. To find an approach to these mechanical phenomena, we considered chaos theory, fractal distributions, and connections with traditional science. Many approaches to study were used, and their purely theoretical aspects and metaphysical conclusions should be treated with caution. Since then, several scientific articles have discussed the concept of tensegrity, tensional integrity. This concept of tensional integrity, or tensegrity, undoubtedly sheds light on how organs connect during joint sliding. Americans Buckminster Fuller, an architect, and Kenneth Nelson, a sculptor, worked on macroscopic objects and developed buildings and sculptures with similar characteristics. The basic concept is that applied forces are transmitted to all elements of a construction consisting of long hinged beams connected by pre-stressed elastic cables already under tension. The principle is that an architectural system in which constructions are stabilized through balance between opposing forces of compression and tension thereby allows preserving structural strength, capability for multidirectional movement, and independence from gravity. A structure based on tensegrity is stabilized not by each part's resistance, like a column resisting gravity, but by distributing equilibrium between mechanical stresses throughout the network. Thus, such structures are stable in all directions and don't depend on gravity. These forces stabilize the construction through their equilibrium, but most importantly, all forces act on all elements of the construction, resulting in the slightest increase in tension of one element being transmitted to all others, even the most distant ones. In real applications, the resulting forms are often geometrically regular, triangular, quadrangular, or various degrees of icosahedral. It's important to understand that to fill space, living structures must obey forces that make them take various forms. It would seem that the icosahedron is the optimal form for filling space with minimal filling. In fact, nature often applies self-assembly rules when creating natural triangular, polygonal, or spiral motifs. As recent research has shown, at the molecular level, as in the collagen molecule, amino acids with a long chain folding into a spiral become stable thanks to the balance between hydrogen bond attraction forces and other forces allowing proteins to resist compression. This allows building sequences that behave like flexible joints. At the cellular level, Dr. Ingbert discovered intracellular reinforcement, an internal mesh consisting of fibrillar elements arranged randomly, but providing a fundamental role in restoring the original form after pressure relief. In fact, the discovered pseudo-polygonal construction raises fundamental questions, since it's present at all levels of living matter in the animal and plant world. Considering the importance of physical forces such as osmotic and electromagnetic pressure, it seems this is the preferred construction system that nature invented during evolution. We must pay tribute to Darcy Thompson, the Scottish humanist and biologist of the 19th century. He believed that Darwin's adaptive teleology should be supplemented by including physical and mechanical causality. Convinced that neither chance nor fate can explain the stunning diversity of biological forms, the undeniable unity of organisms, and the limited number of living forms, he included physical forces in a theory accounting for the multitude of natural forms. Intuitively, he came upon the idea that the forces shaping life are similar to each other. He wanted biology not to leave aside physical aspects, to include physical mechanisms to account for both form and growth, and not to be completely focused on teleology. We began to wonder whether these triangular, rectangular, and polygonal forms we filmed could be parts packed into an icosahedron, the basic element of tensegrity, which so closely resembled a three-dimensional vacuole. These highly efficient, flexible, pre-stressed fibrillar architectures, combining high mechanical strength and optimal material economy, thanks to their ability to take various forms, undoubtedly contribute to greater stability and sliding adapted to each other. This presumably improves their metabolism and thereby prolongs life. Surgery feels this pretension every time when making an incision on the skin, when the edges spontaneously separate. And at the end of the intervention, you can also feel how strongly you need to pull the edges together to stitch them when suturing. It seems that this phenomenon is so widespread that we simply take it for granted. We begin to understand the enormous complexity of the mechanism. However, apparently it's insufficient to explain these sliding movements simply as bringing ropes and rigging into accordance with an order predetermined mathematically, even fractally. Other factors need to be considered. First, it seems that fibers, thanks to their nature, have the ability to stretch or contract. Upon closer examination, it turns out that twisted fiber overlays stretch immediately before joint movement begins. This may be the first stage of a form of preparation before processing a movement request. To respond to the direction of applied force, the biomechanics of microfibrils provides resistance through the additional ability of fibers to move around a nodal point encompassing another fiber. Finally, collagen 4 and 6 apparently can separate and then reconnect as if nothing happened. It seems they can also split into several parts, like the mythological monster with Hydra's head, responding to any requests for mechanical displacement in space. This ability of matter to self-organize particularly suggests infinite potential for movement. Nevertheless, the collagen framework plays any role in this staging. Constant lubrication of joints by glycans through these transparent beads with drops resembling morning dew, omnipresent, as well as fluid migration inside fibers, suggests that hydraulic mechanisms should play a decisive role here. The appearance of bubbles during rupture, when the structure encounters the atmosphere or during tension, means the existence of equilibrium pressure. Moreover, the importance of these glyceramino-glycols, which have different concentrations depending on location in the body, should not be overlooked. Regardless of the final answer, the flexibility of these tissues is determined by the mechanism of one of the main driving forces, which may be tensegrity. But still, tensegrity can't explain everything here. This is a complex sliding system that maintains construction and maintains mobility, and thereby changes according to circumstances and obeys natural laws of change. This happens either as a result of overload, as in obesity, where the corners stretch to diapsids, or as, for example, in tenosynovitis, where fibers tear away from a tendon. But this can also happen with withering, which accompanies weight loss or skin aging. This collapse of our collagen matrix doesn't happen at a constant rate, but this process is inevitable, since gravity gradually takes its toll. The tension of fibers that maintains the tone of our tissues gradually weakens over time, and again entropy emerges as the absolute winner. Whatever the finale of the story, understanding the mechanical and biomolecular characteristics of this flexible communication tissue will one day allow us to understand the full meaning of the healing process, reconstruct living structures, and maybe, who knows, fulfill humanity's eternal dream, slow down the influence of time. Translated and voiced by Alexander Pascoe, YouTube channel, Proper Bodybuilding.